Hello everyone, and welcome to CRAMSURG, Clinical Research Appraisal and Methodology for Surgical Trainees, where we pick a paper fresh from the press on a hot general surgical topic. We review it for you, we present it for you, we critique its methodology for you, and provide top-of-the-field expert opinion and teaching on research appraisal and methodology. My name is Gio Perrin, and together with Professor Saba Balasubramanian and Maria Digby, we bring you CRAMSURG from the wonderful region of the Yorkshire and the Humber.
We've had a few talks on randomized controlled trials, and we have previously highlighted the challenges that surgical RCTs face in not just designing the RCT, conducting the RCT, but also interpreting the results and making it generalizable, making the results of the RCTs that you do externally valid to a much wider group of surgeons out there in the community whose practices might vary, whose local facilities might vary, and whose patients' complexities might vary as well. So surgical RCTs are a particular problem, very dissimilar to medical RCTs, as you would imagine. And I was going to illustrate the difficulties in surgical RCTs using an example in my practice. There is a condition called post-surgical hypoparathyroidism, a lot of you may have heard of, where the thyroid surgeon, while doing a total thyroidectomy, can inadvertently damage the parathyroid glands, damage their blood supply, inadvertently remove them, and the patient has this condition called post-surgical hypoparathyroidism. And there's been a lot of research ongoing, particularly in the last 20 years, on how to reduce the risk of this problem. Lots of short-term issues, lots of long-term morbidity. In fact, there's also an impact on mortality. If you are hypoparathyroid after thyroid surgery, in the long term, your mortality is slightly higher compared to a population who did not get hypoparathyroidism. So one of the proposed solutions has been to incorporate fluorescent imaging of the parathyroid glands during the operation. And fluorescent imaging is considered to help by reducing the likelihood of the parathyroid glands being inadvertently damaged or removed. And also, fluorescent imaging helps in identifying the blood supply to the parathyroid glands. And by identifying the blood supply, you're able to preserve the viability of the parathyroid glands. So on the top, you see how a small snippet showing how fluorescent imaging helps you see the parathyroid glands. And in the bottom is a little video of how you can assess perfusion by injecting ICG indocyanine green during surgery, and you can see the perfusion of these parathyroid glands. So I've been involved for some years in this multi-centered randomized controlled trial, and that we have carried out in the UK, and this has recently been concluded, and this is called the NIFTY trial, Near Infrared Fluorescent Imaging to Prevent Post-Surgical Hypoparathyroidism. And because surgeons do thyroid surgery very differently, we initially thought we should do what we call a process evaluation, where thyroidectomies were observed by qualitative researchers who also did surgeon interviews, and we did some surveys to establish the variability in practice in the UK and to get a consensus on what steps of the thyroid operation should be mandatory and what steps should be optional. We put it to an expert panel, and then we developed a manual for the trial to stipulate which steps of thyroidectomy should be documented and which should be mandated and which should be optional in both the standard and the experimental arms of the trial. Then we set out to do a seamless phase 2, phase 3 design, whereby we randomized patients initially into the phase 2 arm of the trial. When I say randomized, we had one group of patients who underwent the standard operation, and the other group underwent in the experimental arm, underwent the operation under the guidance of fluorescent imaging. And we looked at some outcomes. Our key outcome was 6-month hypoparathyroidism rate. This trial went through phase 2 and then had to be concluded. we didn't go through to phase 3 because we found that there was no difference between the two arms, and therefore the new intervention was considered futile. Now, when we assess, or when we have to assess interventions like these, surgical interventions particularly, these are complex interventions, as you can imagine. It'd be easier for you to understand why I say they're complex if you've assisted or done thyroid surgery, but I'll try and explain as we go along. Now, what are complex interventions? The complex interventions are interventions that have any one of the different sort of characteristics that you can see on the screen. They can have a number of interacting components. They can have a number of different sort of behaviors, which means that which refer to the different ways in which the operation is done in the standard or the interventional arm. There could be a number of groups at which the intervention is targeted. There could be a number of different outcomes and differences in how the outcomes are defined. There could be flexibility in how the intervention is actually implemented. So if you have any one of these issues with your intervention, that becomes a complex intervention. There are other ways in which complex interventions have been defined, essentially non-standard interventions, having different forms in different contexts, have multiple interactive components, and non-linear causal pathways. Again, I'll try and explain this as I go along. Now, surgeons would typically ask, well, why don't we just keep things simple? And you can make this argument for any intervention in surgery. And you could keep it simple by saying, well, I'll ask a very simple question, such as, can fluorescent imaging be effective in reducing long-term hyperparathyroidism in thyroid surgery? And design a simple enough trial to just get to the bottom of this question. However, the answer that you get to the question, to the simple question you've asked, may not be very useful to a wider community of thyroid surgeons if the surgeons have different perspectives on a number of different things. Surgeons could have different perspectives on what actually fluorescent imaging means. Is it an image-based system? Is it a probe-based system? Is it autofluorescence? Is it ICG fluorescence or some other dye? How you implement fluorescent imaging? Do you do it right at the start of the operation, throughout the operation, at the end of the operation? How do you interact with the fluorescent imaging and what it shows you? What is the interaction between how you change your operation in response to what the imaging shows you? And what about the outcomes? Is long-term hyperparathyroidism the only outcome? Is short-term hyperparathyroidism important? Would you be interested in other outcomes such as operating time? Does it help you conclude the operation quicker? Does it help you train residents quicker? So it's very difficult to keep things simple in a complex intervention. The next question surgeons would often ask is, do you really need randomized controlled trials to evaluate these things? Can we not go with our gut feeling, with plausibility, with our experience and tradition? Now, the answer to that is we do need to assess surgical interventions as we would need to assess any medicine or any drug. And that's the basis of evidence-based practice. And we do need to be able to answer questions such as, are they effective? What are the risks? What are the costs? And do the benefits outweigh the risks involved? And we need to be clear on whether this new intervention or technology is better compared to the standard. And just saying better isn't necessarily complete. You need to be clear on better for who. Is it better for the surgeon in some way, better for training, better for patients, better in what way and to what extent? Now, in some surgical procedures, the benefit to risk ratio is pretty obvious, and the intervention has clear plausibility or a biological rationale. For example, you're not going to be asking for evidence to jump off an aeroplane with a parachute. You'll just use the parachute. Similarly, in surgery, you've got some interventions such as overrunning a bleeding duodenal ulcer, fixing a fracture, doing a laparotomy for peritonitis. The benefit to risk ratio is so obvious then you're not going to be saying, oh, I need a randomized controlled trial to assess this intervention. But many other surgical interventions have a very marginal benefit, sometimes no benefit, but have still been introduced into routine practice. I've highlighted some examples. I've listed some examples. You may have a problem with one or more of these, but that's up for debate. Like, for example, RFA of liver metastasis, debulking surgery in advanced renal cancer, keyhole operation on the thyroid gland. But these interventions have been introduced into practice by trial and error, often reported by centers of excellence, where some experts have done it, have had good results, and then they get disseminated, sometimes pushed by the industry, sometimes wanted by the patient, sometimes pushed by enthusiastic surgeons. Some good examples. They could be success stories like laparoscopic cholecystectomy. You're probably aware that we started doing laparoscopic cholecystectomy and gradually replaced open cholecystectomy before there was clear evidence on the basis of randomized controlled trial. So this is purely an introduction into practice by trial and error. Some examples, which are not so successful, that I've listed on this slide, are gastrojejunostomy without vagotomy. That was a disaster. Jejunal bypass for obesity became very popular for a few years and then died out. So clearly, in some instances, you really don't need randomized controlled trials, but there are many, many, many other instances where you need them. So if you're coming back to our example of fluorescent imaging in thyroid surgery, and you're asking the question whether fluorescent imaging is better than standard surgery in reducing parathyroid damage and reducing long-term hypoparathyroidism, a number of people have said that, well, it clearly works because the parathyroid glands fluoresce at particular wavelengths. You can visualize this fluorescence at surgery, and if you see the parathyroid glands better, then you're not going to damage them or remove them inadvertently. And finally, people say, well, if you can demonstrate provision of these glands by injecting ICG and looking at the blood vessels going to the parathyroid gland, you're able to assess viability. So because it is so obvious and logical that using fluorescent imaging can potentially reduce parathyroid damage, why do you need randomized controlled trials? What is the problem? Why not you just get on and use the technique? For every problem, if there's an easy solution, it is very often that the solution is neat, plausible, but wrong. So you might have a solution that appears very logical and intuitive, but you will see in medicine and healthcare that these so-called plausible solutions often don't work in the real world, are not effective. There are many examples, and many people would make the point that without rigorous evaluation, a number of surgical technologies and interventions just turn out to be toys for the surgeons to play with or gimmicks. You can see on the screen, I've got a picture of the angel chicks prosthesis. Some of you may have heard of it, used to allegedly reduce the risk of reflux disease and for hiatal hernias, no longer used because of terrible complications that follow. You may or may not have heard of the bypass between the external carotid and internal carotid circulations that was touted as a way of preventing or treating stroke. So there are lots and lots of examples of surgical technology that have fallen by the wayside. And there's a guy called Scott, who's a gynecologist, who very eloquently described these kinds of technologies as the Scott's parabola, where he says it all starts off with a promising idea. There are some initial case reports, and people get very excited, particularly surgeons. And then surgical pressure, media pressure, patient pressure, these technologies get introduced into standard practice. And after a while, doubts start to creep in. You get some major complications, and these devices and technologies fall into disrepute. However, there are some other technologies that have not been through randomized controlled trials, but have still withstood the test of time. And in my sphere of practice, nerve monitoring, intraoperative nerve monitoring of the recurrent laryngeal nerves is one good example, where after an initial peak of inflated expectations, as you would call it, people get a bit disillusioned, a bit deflated, and then after a while they start to realize that, well, it's not the panacea, it won't completely avoid nerve injury, but it's got a role in thyroid surgery. So I hope I've emphasized and explained that we do need to evaluate complex interventions. There are a number of factors that drive innovation in surgery, and one of the most important underlying factors is a surgeon's desire to innovate and improve patient outcomes. Of course, there are other factors, some of which are industry-driven, some are patient-driven, and some are simply the need to increase efficiency and reduce costs of your treatment pathways. But the innovation has to be assessed for reasons that I've just explained, and that assessment should really be on par with medical innovation, because this is what forms the basis of the so-called evidence-based surgical practice. But the problem with the surgical innovation and the assessment of the innovation is that it's not as straightforward as medical practice. Well, assuming if you believe that assessment of drugs is straightforward. But the particular challenges of assessing surgical technology has led a number of experts to come together and propose what they refer to as the ideal framework. Now, I think we've got a set of slides on the ideal framework, so I won't dwell on it, except to just highlight that there are these phases in the ideal framework that a particular technology needs to go through, starting off with the idea stage, and then the development stage, the exploration stage, followed by the assessment stage and the long-term monitoring stage. And if you just pick out phase 3, which is the assessment phase of the ideal framework, this is when a technology is gaining widespread acceptance and is being considered by many people to be a replacement for current treatment. And this is where you start to think about doing randomized controlled trials of the technology in comparison with the standard treatment. And randomized controlled trials, as you all know, sit right near the top of the evidence-based hierarchy that underpins our practice of evidence-based medicine. Now, coming back to fluorescent imaging. Like I said, there are some specific difficulties in assessing surgical interventions. And the first is that in a complex intervention, and I've explained what complex interventions are, where there are more than one component, and there's some flexibility in how you actually implement the intervention, and that there are interactions between the components of the intervention and the way you do your operation. Also, in surgery, there are lots of confounding variables. There could be differences in how experienced the surgeon is, what the surgical volume is, what the influence of other members of the team, like the anesthetists and the nurses are, and how the patient is cared for both preoperatively and postoperatively. There's also the learning curve and the impact of the learning curve on the outcomes. Your learning curve could be with your standard thyroid surgery, and you also have a learning curve in how you use the technology. The other big problem with many surgical technologies are that the technology itself improves. So by the time you start a randomized controlled trial, especially a multi-centered one, by the time you get funding, design and run the trial and wait for medium to long-term outcomes, it'll be several years down the line. And the technology would have changed dramatically and may not be similar to the technology that you've actually spent years evaluating. Which then leads to the question as to when do you actually assess the technology? How long do you wait for the technology to ripen, to mature before you actually assess it? And that leads us to the so-called Buxton's law, which says that it's always too early to assess the technology until it is suddenly too late. When we say too late, we mean that the technology may have just got completely embedded into practice and you're not going to change that, or that the technology has been used and discarded, by which time it's too late to then assess it in an RCT. So there are some specific properties of complex interventions, and I'm going to go through these properties They might be a little bit complicated and difficult to understand if you're hearing about this for the first time, but I think it'll provide you some useful insight into these problems, especially if you're going to be involved in doing randomized controlled trials or you're going to be reading papers on randomized controlled trials. I hope this will be useful. The first thing is that often there are multiple components to the intervention, to the complex intervention. Like I've already explained, the different ways in which fluorescent imaging can be construed. There's this autofluorescence, ICG fluorescence, differences in times of use, differences in how you interpret it, and how you make decisions based on what the fluorescent imaging actually shows you. There's another property with complex interventions called non-linearity, which essentially refers to, with this example in mind, you can, it's not an all or none thing, the fluorescence. And if you have some fluorescence or a lot of fluorescence, they can be dealt with in the same way. And the decisions on what to do with a gland that is not fluorescent at all, which you might consider non-viable, it really depends on what the other glands are like. So it doesn't flow in a very straightforward manner as to what anyone would do if faced with a non-viable gland. There's another property called emergence, which refers to unanticipated events arising without intention. For example, you might detect fluorescence in a lesion in the central neck away from the parathyroid, and that could turn out to be a lymph node metastasis from thyroid cancer. And if you pick that up because you use fluorescent imaging, that'll completely change the extent of the operation that you're planning to do. There's another property called feedback. Basically, that's an observation that reinforces, balances, or diminishes another factor. So, for example, if you're using fluorescent imaging and you identify a really small inaccessible gland on one side of the neck, it might prompt you to change your technique on the other side so you can identify the gland on the other side in a much better or an earlier manner. There's something called adaptation, which refers to a change in behavior in response to an intervention. So if you're concerned about parathyroid viability using fluorescent imaging, then you might change the extent of surgery. And there's another property called path dependence, which refers to specific paths or strategies you might take because of some specific complex local reasons. For example, if you have a non-viable parathyroid or a parathyroid that might be a bit ischemic, in some centers, you might, before you reimplant it, you might want to do frozen section to make sure it's not cancer tissue. In some other centers, you might not do it because you don't have facilities for frozen section. And finally, actually, most importantly, there's human agency as a property of a complex intervention, which essentially refers to your values, your beliefs, skills, and goals as a surgeon, and how much trust you place on the technology, and how much you would use the technology to make your decisions, or how much you would just rely on your own instinct and what you see with your naked eye. So these kinds of problems are not unique to surgery. So if you look at complex public health interventions, they've got very similar problems. And they call these trials realist randomized controlled trials, where they say that complex intervention RCTs take account of the complex nature of the intervention to understand what works, for whom it works, and under what circumstances. However, there are other people that say realist RCTs never work, and the phrase realist RCT is an oxymoron. So there's this debate going on in the literature as to how complex interventions should be assessed in using the scientific methods or the study designs we currently have. So coming back to the research question, I said, initially that the question for us was whether fluorescent imaging is better than standard thyroid surgery in reducing hypoparathyroidism. The important thing to keep in mind is that, and this is really important for complex interventions, is that the question underlying your study of the complex intervention should be specific, focused, and clear. And you've got to keep in mind that there might be other research questions that might be equally valid. There might be another researcher who says, well, actually, I'll use fluorescent imaging, but my question is, does it help thyroid surgical training? Or somebody else might ask, does fluorescent imaging actually help me remove more lymph nodes in thyroid cancer surgery? Or they might say, I'm not really interested in long-term rates, I'm interested in short-term rates, and so on. And again, the technology in question might have different versions, might have different types. I've already mentioned that there are different types of fluorescent imaging, autofluorescent imaging, and fluorescent imaging using specific dyes. And there are these image-based systems and probe-based systems, which probably will be a little bit complicated if you've not heard of this before. So going back to looking at fluorescent imaging in thyroid surgery through the scope of the ideal framework, the first question is, is the treatment described a complex intervention? Yes, it is. The second is, have you got preclinical studies being performed to assess harm? Yes, in the context of fluorescent imaging. Then the question would be, have early human studies and technology modifications been done? The answer is yes. And have they been replicated and quality measures have been defined? The answer is yes. And finally, have you done randomized controlled trials? So having been through all of this processes in the ideal framework, you then do a randomized controlled trial, which is what we've done. And what do you then do if the randomized controlled trial results are equivocal? In other words, there's no difference in long-term hypopathorism rates. What does it actually mean? Does it mean that the technology is useless? You might as well get on and do your standard operation and forget about the fluorescent imaging. Could it be that your surgery has got better, you're now operating differently, or the technology has changed, and before you condemn the technology, you have to think as to whether maybe you trialed an old version of this, maybe the newer versions might give you a different result in another trial? Or do you ask whether you've asked the right question? You need to ask a very clear, focused, and defined question if you're planning an RCT. And if you've said, oh, my question is whether this improves long-term hyperparathyroidism rates, what about the other questions that you could ask and get a different answer with regards to the technology? And then if you think that the fluorescent imaging really works in your hands, but then the RCT has given you negative results, then you're also wondering whether randomized controlled trials are the right tool in assessing surgical technology. Or maybe there are some design issues in your RCTs. And the design issues could be simple things like maybe the surgeons haven't got used to the technology and they're not using it right. Maybe the technology is used in so many different ways by the different surgeons in the trial, so you've not really standardized it properly. And then finally, maybe there's a Hawthorne effect, which simply refers to the fact that surgeons know they are in the trial. So regardless of whether you're in the standard arm or the technology arm, they're taking great care and are going to great lengths to reduce the risk of this complication. Now, this particular trial, the NIFTY trial, like I said before, has closed after phase 2 because of negative results, because of futility. So what are the implications of this? The first thing is maybe that there was a type 1 error in the trial. Maybe there is a real impact of the technology, but the trial hasn't picked up on it. And what can then happen is you lose the opportunity to assess other outcomes that can be influenced by the technology. Maybe the technology is good for training, reduces operating time, reduces short-term outcomes, short-term morbidity, but you haven't picked that up because the trial was designed to look at a specific long-term outcome. Having a negative result in such a big trial can dampen further research. People might say, fine, that's it. Fluorescent imaging is of no use in thyroid surgery, regardless of the type of fluorescent imaging, regardless of how sophisticated your technology has now become. And then you might also make draw the conclusion that if it doesn't help in routine thyroid surgery, it may not help in complex thyroid surgery. It may not help in settings where thyroid surgical volume is not that high. You've done a trial in high volume centers. Maybe the technology is not needed by the experts. Maybe the technology is needed in people who don't do much thyroid surgery, who do the occasional operation. And then sometimes, as we're finding with fluorescent imaging, I think that it is particularly useful in difficult operations. But if a trial of this nature has shown that it is not useful in the standard operation, then you're not going to get funding to use it in the complex or high-risk operations. So finally, I think I've got just one or two more slides. My personal experience, what's happened with my hypoparathyroidism rates. So over 12 years, I found that my hypoparathyroidism rates were fairly stable, exactly at 5%. Neither my experience nor my volume helped me reduce my rates. And in the last 2 years, my rates have dropped from around 5% to virtually 0.5%. And this in the last 2 years, I've been getting involved in the trial. We've been recruiting patients into the trial. Now, you could say, I've been using the technology. That's why my rates have gone down. However, when I looked at my data, the rates were the same regardless of whether I use the technology or not. You could say I'm getting involved in the trial. So there's this protocol effect of the randomized control trial, or the Hawthorne effect, the effect that I'm being observed. But the rates were the same regardless of whether the patients took part in the trial or didn't take part in the trial. So even if they took part in the trial, my rates were the same regardless of whether they had fluorescent imaging or no fluorescent imaging. So what's happening? I don't know what the answer is, but I have changed behavior. I've changed how I do a thyroidectomy now since I started using the fluorescent imaging. I've gained a lot more knowledge on how the blood vessels to the parathyroid glands are related to the thyroid capsule. I've also been, because I've been getting involved in the trial, been taking very detailed notes of the locations of the parathyroid glands, the blood supply, and how I do the operation, and willingly or unwillingly been involved more in reflective practice. So it could be that these have changed my results. But that's only happened because we started getting interested in the technology and getting enrolled or enrolling our patients in the trial. So, in summary, I was hoping to illustrate the challenges of assessing surgical technology with randomized controlled trials. And I wanted to emphasize that many surgical trials assess interventions which are complex. And I hope I've explained to you what a complex intervention is with an example. Also, I hope that you understand why complex interventions are difficult to evaluate for effectiveness, why surgical trials struggle to establish effectiveness. So it's not because surgeons don't know how to do randomized control trials, or it's not because surgeons don't do randomized control trials, but because these are particularly difficult challenges. And the RCT approach may not always work, and we do have many interventions in standard surgical practice that have not been through RCTs, unlike in medicine. But it's the best of what we currently have. And that's why it's really important to interpret results of RCTs, even the negative RCTs, with caution and pay attention to good quality observational trials. I think that concludes my talk. Thank you very much.
Thank you everyone for tuning in and listening. Until next time, keep running your life with our surgical podcast.